Hidden Markov models are used for different applications in biological domains, such as multiple sequence alignment, annotation of data, gene expression data, gene finding, and so on. In this module, I have given an example of gene finding using hidden Markov models, and I will explain different steps involved with the help of an example. So number one, what is gene finding and what is its different objectives? So basically, idea of gene finding is to find out coding and non-coding regions in a given DNA nucleotide sequence. And what are advantages? Advantages is, of course, number one advantage is of annotation, that we want to find out which part of a specific DNA sequence is useful and which one is not useful. Since there is a huge increase in genomic data, so this DNA finding or annotation becomes very useful. Similarly, we want to have some insight or some light on the inner mechanisms involved in genes. So those mechanisms can be useful while transcription or translation of our DNA data. So for example, what I have taken here is, suppose I have a DNA nucleotide sequence. There are three things that are present in that sequence. Number one is introns, exons, and our 5' splice site. So moving on, what we want to find out is, we want to find out in that sequence where is the exon region, where is the intron region, what is our 5' splice site, and where is intron region ending and exon region starting, and vice versa. So some statistical properties about exons, introns, and 5' splice site. So those statistical properties are basically, whenever we have an exon, we say that it has uniform distribution of our 4 bases. So by uniform distribution, we mean that A, C, T or G, all of them have 25% probabilities that it can start from any of these. So it can start from A, C, T, G, anything, and all of them have 25% equal chances. For introns, on the other hand, A and T have high probability. They have 40% probability individually, and then the remaining C and G, they both have only 10% probability. And then for 5' splice site, highest probability is for G, which is 95%, and the remaining 5% is for A site or A base. So whenever we talk about HMM, what we need for starting any HMM, we need states. And in our particular state, we can say we will have 3 states. One for intron, one for exon, and one for our 5' splice site. So based on these sites, what we will try to find out, what are the emission properties and what are the transition probabilities. So I come to the figure of this HMM model, which is manufactured or generated for our particular example, and that this thing presents information is that we have 3 sites, E for exons, 5' splice site, and then introns. So these are our 3 states. For each of these states, we have these emission properties which are shown here. So these emission properties for exons, we have 0.25 probability for A, C, T, G. For all the bases, it's equal. For 5' splice, we have high probability of G only, which is 0.95, and remaining 0.05 is for A only, and others are 0. For introns, we have this emission probability table, which is shown here. So other thing is our transition probabilities. So we have shown 2 more states here, which is start here and the other one is end. So from start, it will always go to the exon, which is 1 probability, and this state transition is from start to the exon site. From exon, it will either go to its emission probabilities, or there is a very high chance, this 0.9 probability, that exon will move towards exon. So state transition will be between exon and exon site itself. And 0.1 is for exon to 5' splice site. And then from 5' splice site, there is no chance that it will come back to itself, because it is just a division state. And what will happen, it will always move towards intron side, which is shown by 1 probability. Once it is in intron, then again there is a high chances that it will remain its own state, which is 0.9, or it will move towards end side, which is 0.1. So we are generating two sorts of information. Number one, what is the observed sequence, and number two, underlying state path, कि कौन सा state path है जो हम follow करेंगे to generate this sort of data. Structure wise, what we said that HMM has S observed sequence, then pi state path, parameters which is shown by theta, and probability which is shown by P S of pi given HMM theta. Basically it would be the product of all the emission plus transition probabilities. So it will give us our complete probability of a hidden Markov model. So for this purpose, what we say that okay, here I have given an example. So this example basically shows that there are 26 total sequences, basically pairs in this sequence. So this information, there are basically 26 number of bases. So for this 26 bases, there would be 26 emissions, so which is shown here. And since there is a one transition for the last slide, so there would be total 27 transitions which are shown here. So what I have shown here is that for a state path, one state path for this data can be number one, these exons up till here, then we have supply side, and then we have introns. And based on this thing, we have calculated our information log of P, this thing, which comes out minus 141.22. So what we are doing is we are just multiplying all the 27 transition probabilities for these states and then 26 emission probabilities, and when we multiply these things together, we get some information, and then we take log of that information. Why we are taking log? To make it in some measurable unit, because these multiplications will result in a very high value. To keep it in some measurable value, we are applying log, and this value comes out minus 41.22. So okay, in previous page I have just shown one value, which one was when we say that this is the state path. So is this the only path? No, answer is no. There can be many other paths. For example, if I take the first occurrence of G, it becomes this path. So first occurrence of G in this symbol, it is occurring here in this place, sorry, G. So what happens if I say that okay, so all the bases before this G, they are exons, and after this introns, so this would be the result. And when I take log value of this symbol, it becomes 43.90. And similarly, these are all other places, such as, so this one is G is taken here, and then G is here, and G here, G here. So I have shown different observations which can be made for this particular sequence. And here I have given values of all this information. So what we have shown here, that we will pick the observation which has the lowest value, which is minus 41.22, or at the negative terms it is the highest value which is available. So which has the maximum value of log P, we will pick that sequence or observed sequence. Confidence score. We said that fifth occurrence of G is the right choice. Is this the right choice? How we will define that this is the right choice? For that thing, we have to calculate the probability of nucleotide I, and that we say that this nucleotide I was emitted by state K, and it is basically sum of the probabilities of all the state parts that use state K to generate our observed sequence, that is I, and normalized by sum of all observed paths which are given in our HMM. What does this mean? For example, what we have shown here is that when we have these paths, so for our G, which is at the fifth location, we are showing this thing. So it has the 46% of score, which is the highest, and the second highest is this one, which is shown at the sixth place of G, which is 28. So by this score, we say that the best path is this G. So is this HMM can be extended for other things? Yes, answer is yes. We can take combination of other bases also. These bases at the moment we were taking only 4. We can add this up to 5 or 6 symbols. Similarly, we can add different states for HMM. For example, this was only for 3 states. It can be extended to any states, let's say for 4, 5, 6 and so on. So we can easily conclude that there are no limitation of possibilities when we are finding genes through a hidden Markov model.